Can you hear me? Yeah. So I was amused. First of all, it's an honor, a pleasure to be here, and I'm very excited about this conference. Hearing about it and then reading the questions that were posed were all, for me, very exciting, and I'm really looking forward to the interactions that we're going to have, not just the talks, but also the conversations, and hopefully digging deeply into some of these problems. And I was amused listening first to Randy and then David's presentations, because basically they covered most of what I wanted to talk about. And I do have, I'm going to try to pose answers to some of the questions, and they're going to go across the range of the topics that are presented here. And so I have to start by asking, are there any questions? So let me get this up on the screen, hopefully. And so I'm going to emphasize some new ideas that are coming out of Birdsong research, talking about results that go all the way from ion channels to behavior through some interesting levels of processing. And we do have some answers now, I believe, and I think we all know the experience that good science generates more questions than it answers, and that's certainly the case here. So today I'm going to focus just on one species, the zebra finch, and I'm going to largely ignore the part of birdsong learning that you probably have knowledge about and experience in, which is the developmental process by which birds acquire their song or songs, depending on species, which is a very, very rich part of our field. And in some sense, I can go back beyond even 1960. I apologize right now, I forget the name of the then president of the American Physical Society, who in his presidential address lamented the fact that comparative psychologists had gone out searching for general theories and had, as he described it, essentially disappeared because they found one animal, the white rat, and stopped doing comparative work. And there's a danger of that in birdsong as well, where there's a vast amount of work done on this one species, and the richness of the other 4,999 species is missing in that work. So I'll try to touch a little bit upon that today, but I'll really focus on the zebrafinch. It does come with, these animals do come with enormous technical advantages. And so, and here, this is just some graphics to describe this. So I'm focusing now on the song of the adult bird, and I just want to introduce you to a few concepts so we have that clear. This is a frequency versus time representation. This is a sound spectrograph. I presume you're familiar with this. These are introductory notes, and then here is a sequence. In this particular bird, it's a bit unusual. It includes the introductory note and then a sequence of syllables that form what we call a motif. And you can see as the bird sings, he goes, and those sounds are very, very precisely produced. And in fact, the males sing and the females choose the male in part based on how precise the male can produce his song. And so all of the song itself sounds horrible, even worse than what I just produced. These are champion species. This is an example of a champion species, an animal that does something about as well or better than most animals in the animal kingdom. And so it's a good species to study if you want to look at how the nervous system solves a particular problem at the extreme of what it can do. And so that can be useful. That can allow us to see things structures in the brain that we otherwise wouldn't be able to see. And you can see here are a bunch of songs from different birds. This is taken from a data set that Ofer Chernikowski contributed to. And you can see these introductory notes. Virtually all zebra finches, not all, but virtually all zebra finches sing them. So it's a series of introductory notes, speeding up, and then the motifs. The behavior is much more complicated than that, but that's the cartoon of the behavior that we're going to talk about today. Okay. So our work starts in a surprising place. We're going to look at the neurons in the song system that project to the basal ganglia, part of the song system, part of the basal ganglia. And we're going to, instead of doing recordings from awake behaving animals, which is what I've certainly done much of my life and many people in the field do, instead we're going to look at recordings in a brain slice preparation. We're going to do whole cell patch recordings. And truly, this is the royal we. All this work was done by my former postdoc and now assistant professor, Arij Da. Now, these cells in this structure HVC that you'll be hearing more about in my talk and in Mike Wong's talk to follow, these cells that project at the basal ganglia are very, very distinct. And one can pick them out even in the slice preparation with great confidence. So in these experiments, we retrogradely labeled them. We also intracellularly filled them. And their physiological properties themselves are very distinct. So Arij recorded from a pretty vast number of cells from animals. It's a plausible argument that every single one of these 370 indeed was an X-projecting cell. If we made mistakes, it was a very, very small number. That's a starting point. So here basically are the data. We're going to delve in some depth about this, but right in front of you here is a very startling result. So these are recordings from 3 birds. This is bird 1, bird 2, bird 3. And you might think you're looking at the responses, the intracellular responses to a current injection from one cell, but in fact you're looking at it from 8 different cells recorded in this one bird. And what you can see is that each one of these cells emitted either 4 or 5 spikes, and there's a similarity in the train of spikes, the timing of the train of spikes emitted by each of these cells. And that's a surprising result. That's not the way it's supposed to be. Now, we did a few things to help us out here. Every single cell was held to minus 70 millivolts, so when the resting potential upon break-in was slightly lower than minus 70 millivolts, we lowered it to minus 70 millivolts. And in the majority of cases, in fact, when the resting potential was below minus 70 millivolts, we brought it to minus 70 millivolts and then made a current injection 100 picoamps, 200 milliseconds. That's the so-called canonical current injection. And you see that all the cells in this one bird are rather similar to each other. And all the cells in the second bird are rather similar to each other, but different from the first. And that's the same thing for this one. Now,
do you have synaptic blockers in the bath at this point?
So we've done many, many things. Give me a few moments, and all the concerns about, so we've seen this with synaptic blockers, to be sure. And all the concerns about uncontrolled variation, I will put to rest. So just give me a few moments. Okay? So we can characterize these data in a variety of ways. So for instance, we can plot the spike frequency of a given train of spikes against the first inner spike interval, and those two should be related, and they are, but the circles here, just a few examples, show all the cells from a given bird, and you see that they always cluster. Not perfectly, but there's clustering of all the cells from a given bird, and the next bird sits somewhere else in this trace. And again, that's a surprising result. Why should it be that way? That's not the typical description. Okay? And of course, then we can reduce the data set and find a simple model that describes it. And it's highly, highly statistically significant. Okay? Now we can also then look at the actual shape of the spike waveforms. These are places neuroscientists normally don't go. What information is there in the shape of the spike waveform? Well, we'll talk about that in a moment. So what we're doing here is just focusing on the very first spike in the train of spikes in response to the canonical stimulus injection. Again, these are whole cell patch recordings into the somata of these basal ganglia projecting cells. And here are 10 cells where the first spikes are overlaid. And they're very, very similar. The shape is very, very similar from these 10 different cells. And here are 9 cells from another bird. And again, the shapes are very, very similar. And here from the third bird, 8 cells, the shapes are very, very similar. But this is different than this, and it's different than that. So that too is a surprising result. And here are the average from 8 birds where we had a lot of these recordings. And again, you can see that in each one of the birds, the shape of these first spike waveforms are different, one from the next from the next. So you won't find this in Candel and Schwartz or whatever the current authorship is. Something peculiar is going on. So what is that thing? How do you relate the spike shape and the trains of spikes? We'll see that in a moment. So here we're now focusing on the shape of the spikes to show you the population data. This is the plateau amplitude plotted against the first spike amplitude. And again, the data from individual birds cluster in a small part of the space. Basically, you can take any 2 features of the morphology of the raw data and plot them up, and you'll see this form of clustering. So what's going on? Well, the way to think about this, of course, is the fact that spike shape is important because spike shape is related to the intracellular properties of the cell, and the intracellular properties of the cell help to govern not just the synaptic input, but the intracellular properties of the cell help to govern what information coming into the cell is transmitted to the network through this train of spikes or this burst of spikes that the cell emits. And the way we think about that, of course, is through extended versions of the Hodgkin-Huxley type. So we actually know quite a bit about the intracellular, the ionic currents, the ion currents that give rise to the response of these various cells. And there's an excellent work that Rij Dao, or Rij, the same individual, doing these recordings, did during his graduate days. And so in this particular study, what we did was we focused on, for good reason, I'm happy to describe the depth if you want, we focused on 5 particular ion currents. And we made models, which I'll describe in a moment, by manipulating the magnitudes of these currents and holding the other 60 or 70 parameters in this Hodgkin-Huxley-like formulation constant. And again, I'm happy to talk more about the modeling as you want, as you wish. And so Arish did this initially with manual manipulation of these. And so here are examples of 4 out of the, whatever it is, 70 or so cells that he did, or 100. I forget the exact number, but a large number. Here we're taking the actual biological trace and fitting it to the model. And you can see that the fits are quite good, but of course that's not good enough. We then take that model and see how well it predicts the response to current steps that are different than the canonical one that we fit it to. And we get very good fits. And even when we've applied chaotic currents to the cells, the model did quite a good job of predicting the responses to those chaotic currents. So the model is actually doing pretty well, and just to give you a sense of it, this cell down here, which is the worst of the 4, is pretty close to the average quality of fit. So half of the neurons that were modeled did better than this, and half did worse. So something interesting is going on here, and we can now then address first the potential caveat that by doing manual fitting, perhaps we're just looking in a small part of the overall parameter space. So here, what's been done is we've examined the 5-dimensional space of those ionic current magnitudes by tiling them with over 2 billion points and using a supercomputer examining the model prediction at each of those 2 billion plus points. And so here's a cut through that 5-dimensional space as a function of the error. And here is a Regier's estimate for this one neuron. And obviously the two are very close to each other. And that was the case for many of the cells that we did, and in the few cases where it didn't work, we have a very clear understanding across neuroscience. Okay, so let me tell you a little bit about the effects of the delayed auditory feedback on the singing behavior, because that will allow me to now move into a few comments about a more systems level analysis. So if I asked you to, if I had a setup here, and I put a headphone on you and asked you, and a microphone in front of you, and asked you to come up, hold up a book and read out loud, and you would hear your own voice through the headphones as you read, but your voice would be delayed, in the case of humans, optimally by 200 milliseconds. Many of you would find that extremely difficult to do. Your speech would slow down, and you would start to stutter at the beginnings of phrases. And in fact, among stutterers, peculiarly, giving this delayed auditory feedback can actually be an ameliorative for them. There's something very interesting going on there which we don't know much about, because with all due respect, there is yet no science of stuttering. The NIH adamantly resists studying it in a scientific way, so we don't know that much about it. They should do better, and we can do better, but this is where we are right now. But we can get a zebra finch if we expose that bird to continuous delayed auditory feedback for several days. We can get the bird to stutter. So this bird that sang A, B, C, D, E, sometimes with a note in between, sometimes not, A, B, C, D, E, A, B, C, D, E, after several days may sing A, B, C, D, E, D, 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 D. And he does that even after you remove the delayed auditory feedback for weeks on afterwards. That's the long-term effect. The short-term effect is all about song initiation. This is one of many analyses we have to support this conclusion. So here, what you're seeing is a representation. Each one of these yellow tick marks represents the time when an introductory note occurred. These are the first songs that the bird sang with only one motif. So, and so on and so forth. One motif, two motifs, three motifs. So you see some introductory notes whose timing is not well structured, and then the last introductory note, and the song begins in a very regular time, in a very regular fashion, after that last introductory note. When the bird is now put on delayed auditory feedback, these are now data from, I think, the first 4 hours of reporting for this one particular bird. What you see all of a sudden is, it's not that birds without auditory feedback never do this, but it's much, much more prominent that the bird might start to he gets to that point and he can't continue. He never sings. So we call these zero motif songs. We see this in every single bird where we've given continuous delayed auditory feedback. And there are many other measures in the singing behavior that all point to a problem in initiation. Well, that's pretty interesting, actually, because these basal ganglia neurons project to the basal ganglia. And the basal ganglia, one of the things that's been cited as having a role in initiation of behavior. So this allows us to move to a more systems level set of questions. And so I want to introduce a different way of thinking about the organization of the song system, which is not just a top-down representation, but as one of the points that David talked about, there's also a sort of a bottom-up view of the world. And I'm going to suggest to you, I'll develop in the remaining few minutes, how we can, in fact, identify where a decision to act occurs. And my answer to that question is going to be that the place where a decision to act occurs is in the synchronization of the activity across the system of structures to prepare to, in this case, to make a movement and to maintain that movement. So it's the synchronization of independent or relatively independent pattern generators at multiple levels of the system. And that is supported not only by top-down processing, but also bottom-up processing. Now, in the Birdsong system, bottom-up processing occurs one of two ways, either through auditory feedback or in the brain through the projections not of neurons that represent the activation of the muscles of the vocal organ, the syrinx, but through neurons that control respiration. Because if you speak or if you sing, you have to coordinate respiration with vocal muscular activity. So this raises the question of to what degree can we think about a top-down, to what degree can we think about a distributed and bottom-up representation. So there's really beautiful work done years ago identifying two classes, what then we knew of as two classes of projection neurons, those that project down the motor pathway. These are these basal ganglia projecting neurons, all of which fire very sparsely, one burst or several bursts per song motif. And the timing of these relative to song motifs remains a point of vigorous investigation. And then that's contrasted with the activity of these interneurons, which fire much more tonically throughout song. So because there have been some questions about how information is represented, we did something which hopefully is clarifying to some degree. We recorded from HPC neurons. It's a relatively difficult experiment to do, to record from HPC neurons at the same time we're recording from a particular muscle in the syrinx while the bird sings. And this is this ventral syringeal muscle, which controls sort of the overall structure, amplitude structure of the singing wave. Helps to control. And here what's shown are the spike-triggered averages of the EMGs from this muscle, conditioned on the activity of the HPC neurons. And these are better isolated interneurons, these are multi-unit activity. And what you see is that on average, there is HPC leads the activity in the muscle. Typically, on average, something, I forget what the number was, but maybe the average was only 12.5 milliseconds, which is a surprisingly short amount of time. But in this case, there's a lead. The brain is active first, and the muscles contract later. And this is part of causal chain. If we do the same calculation now on the sound the bird produces, there's also a lead, of course, but the sound happens later in time. So this argues for a causal chain, and it's important to note that these recordings were surely dominated by the activity of the HPC interneurons, the tonically firing neurons. It really remains a point of active and fun conversation about what is the timing of the projection neurons in HPC, and I'm sure Mike will have more to say about that in a few moments. So we can also then take the activity of these interneurons and create a simple model summing up the activity of the interneurons, doing a principal components analysis to reduce the space, and find that we can actually predict the muscle activity quite well through the activity of HPC interneurons during singing. So there's a lot of information about the singing behavior in HPC at the time the animals actually sing. And so, how can we understand that? So here in what I hope are, do I still have a few minutes? Or, yeah, thank you. Randy has said I do, so I'll take that. Thank you. So I want to introduce then a model which helps us understand this to some degree. This is work, especially since Adrian isn't here, it's useful to introduce the work of another computational neuroscientist who's a nonlinear dynamicist and trained as a physicist, worked on sunspots originally, and a wonderful guy I've enjoyed for the collaboration for a number of years, who developed originally from the basis of first principles, starting with Newtonian mechanics and then dealing with nonlinearities of the membranes of the syrinx smashing into the walls or smashing into each other while the bird sings, learning how the movement of the membranes induces pressure fluctuations in the air column going through the syrinx, and developed a set of equations to describe that. And then later did a dynamical systems analysis to understand where hop bifurcations occur, where saddle node and limit cycle bifurcations occur. So structures that will produce, dynamical structures that will produce more tonal sounds, dynamical structures that will produce spectrally rich sounds. And then actually years later, identified the so-called normal form versions of the same equations he had written before, which are very similar to the equations that describe the sounds that humans and other mammals produce through the larynx. So this is the more modern version of the theory. This model is beautiful in that it can describe the sounds from many different species of birds. The basic idea, though, is a very interesting one. You saw the point of the importance of dimensionality reduction. So here, the idea is, imagine that the brain actually has relatively simple commands. And those commands drive a complex nonlinear device, which gives rise to the complexity of birdsong and the variability in birdsong across species frequency. That is the deep idea that Gabo had. And we investigated this by looking at the activity in HPC neurons and asking in what ways is it related to these so-called vocal gestures and the either the onset or offsets of vocal gestures, or maybe the maximal tension or maximal pressure. And in our hands, first looking at sleeping birds and doing acoustic playback, we found that HPC interneurons were nicely characterized by these dynamics, and that was also true of projection neurons. Now, again, in complete fairness, there have been data sets from other labs, including Michael Fee's lab and Mike's lab here, and he'll talk about it, I presume, not finding that result, and so we're in active and enjoyable, I would say, conversation about it. We also did these recordings in singing birds and confirmed our results in singing birds. And it's an interesting conversation to figure out what's going on. So I'm going to end here by pointing out that there's a remarkable result sitting here, which is the timing of the activity of one of these projection neurons or one of these interneurons in HPC is precisely aligned within milliseconds to when the bird actually produces the sound. And quote, that's impossible. It breaks causality. So when we got this result the first time, I went to all my physicist friends, Gabo and other people, and I said, do you guys still believe in causation? And they assured me causation is a good thing. Okay, so that was a good place to be. And so we've thought more about this, and the answer is, I mean, there's not enough time for HPC neuron to be active and for the information to get down to the muscle and the muscle to contract and the sound to be produced with just a few milliseconds swap between the two. So the answer is, again, through synchronization. The idea is that as the animal prepares to sing, these different parts of the brain become active and co-active and synchronized. In fact, you can, even in the master-slave relationship, you can have the slave lagging or leading the master oscillating. There's no constraint about that. You can bring the whole system into temporal synchronization, and that's how you can achieve such a result. And the lag that we saw before in the other recordings may be the information that is leading to this synchronization. So that's the idea for how we might get a result like this. Okay, so don't think this is just true for zebra finches. Here is some. This is actually a zebra finch. This is the first evidence of song replay, which we showed a number of years ago. And here's an example of the activity of another one of these projecting neurons in a different species in a swamp sparrow, and the response of that same neuron when John Prather in Rich Moody's lab played back the song. And look, the timing delay is too short to explain through normal processes. The brain does not represent timing the way we think about it. Oh, information gets into a sensory system, it climbs up into the brain, and now it's delayed by 50 milliseconds relative to the event out there, and you decide to make a movement, and that climbs down to your effectors, and then another 50 milliseconds later you make the movement, and there's this time swap everywhere in the brain. And that's incredibly confusing to think about, and that's not how it works, I believe. And I think these data help to support that there really is synchronization. So how can, causally, how can an auditory response appear at that moment in time? Well, David actually gave the answer what was it, 35 years ago? Some young graduate student was looking at white-crowned sparrows and showed that the neurons integrate over a period of time and make a nonlinear summation and a prediction about what's going to happen in the future. And so this is essentially a predictive response, these auditory responses, and we seem to see that throughout the song system. Okay, so I will stop there. Thank you very much, and point out that I think we are finding something new about mechanisms of neural dynamics in study.
Well, that was a virtuoso tour de force that took us down inside the cells and out to the periphery and into the world and back around and again. My brain is oscillating, synchronizing, desynchronizing, and I'm trying to pull out what I learned. I learned an awful lot. Important points: intracellular events rapidly modify. We don't know what the dials on them are exactly, but an extraordinary result with respect to the question of this session, which is levels of analysis, and then levels of analysis at the circuit level, which has to do with the synchronization. So, some questions that are topical. Maybe we might save the most general question until after Michael talks and we've had some coffee.
Once you said we'd have some coffee, I turned everyone off.
Well,
David, should I use the, can you hear me?
just stand up and shout.
Okay. So it's really fascinating. So the core result you showed in the beginning about the intracellular properties, surely that's not only that nucleus that's happening. How is that playing out in upstream and downstream kind of areas that project into that nucleus and areas that receive input from that nucleus? Do you have any information about how intrinsic properties are kind of matched to the response that you showed?
Right. So the question is, how general are these observations about the intrinsic properties in other parts of the system? And I would extend that to other species. And we don't know the answer to that yet. But there's every reason to believe that there's evidence, that other neurons in that same structure, HBC, also show changes in their intrinsic properties developmentally. So it's very likely to be the case that at least within HBC in zebra finches, to be really critical about it, these observations will take. I think is this still on? I can't quite tell. Yeah. I think an interesting way to approach this, something I'd really love to do, and we've actually started to do, is to bring back the comparative neuroscientists to Birdsong, and to look at this question in other species. So, do we see this in other species that also sing a single song repertoire? What about a bird that sings two different songs? Does it start to mush over, or are there two camps? What about a bird that sings 5 different songs or 20 different songs? It's one of the advantages of the system, can we do that? And I can actually think of very nice ways of doing this in mammals as well. So I think there's a research program that allows us to go forward. We were already surprised. We did not go into this looking for this. This was serendipity. The only thing I can feel some pride of is we didn't ignore weird results. So this is new for us, and hopefully I'll have a better answer for you in a couple of years.
Is the number of bursts explained by the cases of the episode, or do you think they go together functional? You have this set of currents that have to produce this set of spikes in order to make it functional.
Right, so could we...
Repeat the question.
Yeah, I will, yes. Because I can barely hear it, so I'm sure even with my bad hearing, somebody might have that. I think another way to ask the question is, can you reduce that parameter space? Are there some relationships, for instance, between the magnitudes of the various currents and the features of the bursts? There are let me think on that for a moment. There are some birds that have seemingly different songs that seem to overlap to some significant degree in the parameter space. In general, the Hodgkin-Huxley formulation will take a given set of parameters of these ion conductances and give you one dynamics of the system. Surprisingly, that hasn't been explored very much. I mean, we're finding a global minima for any one neuron, and that actually is not a well-known observation thinking about Hodgkin-Huxley dynamics today. That was a surprise to us also. They don't have a good answer for you yet. I think there can be some dimensionality reduction, to be sure, but it does seem like there are, in general, distinct solutions for any given set of parameters.
Well, I have a louder question here, but I think you want to repeat the question. I just want to make sure I understood what I took to be an implication, but I don't think you had said it explicitly. So you're looking at the spike pattern introduced in this in vitro by in a totally artificial means, right? But I think implicit in what you were saying is that the way in which these neurons synchronize their intracellular composition is because that composition translates into stereotype, some kind of probe signal or something, produces spike trains from those neurons that allow one neuron to compare its innards to the innards of another neuron. Did I have that right?
Yeah, I think, again, the question is, how does it get to be that neurons in a population of neurons all share common intrinsic properties? It's not quite the same thing, but how could it be that your heartbeat and my heartbeat could become the same? That's a little strange, right? Well, of course, nervous systems are connected in ways that individual organisms aren't. But yes, our view of how this happens is that the neurons are, the intracellular properties of the neurons are representing the entirety of the animal's behavior, the singing behavior. Right? And the way the animal assesses that is by producing a song, listening to feedback, modifying its behavior. So it's the entire activity of the whole network feeding back onto those neurons, which is the input that is modifying its intracellular properties and representing the error and the variation across those neurons represent the error that the animal is then adjusting. So I want to emphasize, it's not that I don't believe that the synaptic input to these cells isn't important. It's that there's additional information that we tended to ignore in the somatic properties of the cells that may be contributing it computationally in a fundamentally different way, I might add. I didn't say this, right? Synapses get modified. We don't know how that's related to, say, counting or something. But when you modify the intrinsic properties of the cell at the soma, you're modifying how the cell transmits information to the network and whether it even contributes to one particular ensemble of activity. And so it's very different, computationally very different. So this animal, its very regular singing behavior and the regularity of the activity of the projection neurons probably gives rise to very, very similar trains of synaptic input to the different cells, and that helps drive the regularity in the intrinsic properties. But it is, I'd love for someone to give me more insight than I have at the moment as to how this arises. It's a bit of a mystery, to be sure. And frankly, if you ask me, do you really believe that all the X-projecting neurons look the same? Well, no, of course I don't. But that's what we find.
Well, once upon a time, you showed that at least in the tranquilized animal, listening to a song carried out a relatively synchronous effects on a large population of cells. They weren't differentiated. So that there is some common drive seems to be reasonably clear in the system. That's a fine point. But with respect to the general issue, you did also show that this is, in a sense, a labile or flexible or plastic property. You claimed, based on a little bit of the data, that 5 motifs, or some 5.7, whatever that was enough for changes to be detected, suggesting not some gene induction event, but some covalent modifications along the intracellular tail of the potassium channels or some damn thing, along the lines of what's been described perhaps in LTP or in certain Ecclesias synapses and whatnot. In other words, the dials are accessible to the very recent history of inputs.
Yes, yes, yes.
And they, of course, they may also encode longer processes because we know that every time you change one of those things, there's a signal that goes to the nucleus as well. So there may also be long timescale events as well, such as the fact that after delayed auditory feedback, it doesn't reset as fast as the problem comes on.
Yes, that's all correct. That's all correct. One of the things that experimentally that it suggests is that if we could manipulate some of those curves experimentally, we ought to be able to manipulate the song that the bird produces in a predictable fashion. It's not an easy experiment, but it's with modern genetic techniques, it's in the hands of the right collaborator, not me. It's a plausible experiment, and again, it suggests that these cells, every cell perhaps in the brain, has a genetically endowed response to changes in its inputs that may modify its intrinsic properties. That's going much further than the data, but maybe. And it's the regulation of the systematicity or regularity of those synaptic inputs that affects to what degree those cells will express similar intrinsic properties. And once they do, it represents its own version of memory in the nervous system, which is distinct from the way we normally think about mechanisms of learning.
So that last point resonates incredibly strongly for me because we don't have the slightest clue what memory is, and this is a new kind of hypothesis. Anyway, I'd like to thank Dan for a very stimulating kickoff presentation. So let's give him a hand. At this point, I'd like to introduce one of our benefactors. Okay, this conference would not have taken place on the one hand without the hard work of Sarah Pixley in terms of organizational work, but it also wouldn't have taken place without the intellectual environment provided by the Rutgers Center for Cognitive Science and the kind of interdisciplinary cooperation that that center represents. Today we're mostly talking at the intersection between psychology and computer science, but we should not forget that philosophy and linguistics, the other members of the Cognitive Science Center, ultimately can contribute to this conversation when we start talking about higher brain functions. And the Center for Cognitive Science has been the recipient over the years of very generous and perhaps uncompensated support from a succession of deans. So I'm going to introduce Dean Peter Marks, the Executive Dean of the School of Arts and Sciences, who has been a friend to this intellectual enterprise. Peter.
Well, thank you, everyone.
You need a microphone, or you
No,
just
I'm going to speak as loud as I can, because I only have 2 minutes. So, I'm a mathematician by training, and now being an administrator, and in some sense my time is not my own. So, I more or less go where I'm told to go, and I was asked to come over here and say a few words, and I wandered around looking for the room. I finally found what I thought was the correct room, but that was confirmed when I came in and I saw on then Margoliash's slide a couple set of equations for a couple set of oscillators and a songbird, right? So this must have been the right place. Right? And so I'm very, very pleased and privileged to say just a few words of welcome. And to say 2 things. One is that, I'm speaking as an administrator, right? So we have departments for a reason, right? We have a mathematics department, and we have a psychology department, and we have many other departments. And those are good things. But there are many intellectual pursuits that cut across these sort of canonical disciplines. And every university has to find a way to cut across these disciplines to address, really serious, really rapidly evolving scientific questions. And that has been historically and is today one of the roles of the Rutgers University Center for Cognitive Science. This is really fabulous. It's a privilege for me to take credit for things I haven't done. Since deans often take credit for bad things, it is one of our privileges to take credit for things that go well. And I think, at least I hope, this is a 2.5-day symposium has gotten off to a great start. I want to address myself to the younger members of the audience here too, because the leaders here, Randy Galestiel, Dimitri Metaxas, and David Vicario, are giving you an object lesson on how to manage up. Right? So they put me in a just 2 or 3 minute slot before milk and cookies. and the lesson you can learn from this, young people, is that, like lab rats, deans will work for food. But in all seriousness, this is a really wonderful example of the cooperation between 2 departments, computer science and psychology, and all the cross-cutting work of the Center for Cognitive Science. I understand that there are faculty and students from over 20 universities in attendance today, and that's great. We like to think that doing things this way, in this sort of ecumenical, cross-cutting way, is a way to pursue research really at the frontiers of certain kinds of disciplines, this being one of them. And it's one of the ways that universities like Rutgers build their reputation. So it's terrific, and I really want to thank you all for coming. And I'm going to end there so we can have coffee and bagels. So thank you all very much.